Hello, this is a tutorial of Bioinformatics homework number 6. The overall goal of these assignments has been to design a plant vector that can be used to transform into switchgrass to enable it to biodegrade methylmercury in the soil of abandoned gold mine trailings. Last week you identified two proteins from E. coli, MER-A and MER-B, that can be used to detoxify methylmercury to the less toxic elemental mercury. This week you will make a plasmid map that illustrates the position and orientation of the different components on the transformation vector that you will build. The first bits of this assignment are some PubMed investigative searches to find the most suitable transformation vector for this project. There are hundreds of more transformation vectors, and it's best to find something that has been used similarly in the past. The assignment starts you off with one of the first reports of gene transformation into switchgrass, and from there you'll need to follow some reference trails to get a picture or map of the vector. Eventually you'll find a paper by Tingay et al. And look carefully at figure 1 and use the legend to understand the descriptors on the vector map. In order to complete this assignment, you need to understand what the various genetic components of this map are. This vector is also shown on the next page of the assignment, but without the legend. The transformation vector is drawn as a circular plasmid DNA. The vertical lines with single letters above represent restriction sites. You don't need to be concerned about the restriction sites for this assignment. The boxes represent the genetic components that encode various functions on the plasmid. The thin lines represent the DNA sequences that separate individual genes. This vector contained only the tDNA portion of the Agrobacterium TI plasmid. The vir and transfer genes have been moved to a second vector. This is known as a binary vector system because the virulence genes and the tDNA genes are on separate plasmids. For this assignment, we're only concerned about the tDNA portion that's diagrammed in figure 1. The regions marked ORI are origins of replication. There are 2 on this plasmid, so that it can grow both in E. coli and in Agrobacterium. BL and BR are the left and right border sequences, which define the transfer DNA. In the Golden Rice paper, these were called RB and LB. AMP-R and TET-R are antibiotic resistance genes that can be used to select for this plasmid in bacteria. A different selectable marker gene, BAR, is used to select plant cells that have been transformed. There are 3 components to the BAR resistance selection. The first is the coding sequence for the BAR gene. This was obtained from the bacteria Streptococcus and encodes a resistance against glufosinate herbicides. The specific herbicide used in this paper was bialafos. The UBI promoter is from the maize ubiquitin gene and is used to drive transcription of the BAR gene in plant cells. The ubiquitin promoter is expressed in most plant tissues. The arrow designates the start of transcription and the direction of transcription. There is a 3' transcript terminator region that's called NOS because it comes from the Agrobacterium nopaline synthase gene. There's a GUS reporter gene on this plasmid as well. Beta-glucuronidase, or GUS, is an enzyme whose product can be stained blue. This allows investigators to see how a promoter is regulated in different tissues, both in plants and in animals. GUS provides a powerful technology, but is not particularly useful for the bioremediation vector. The GUS gene is regulated by the ACT promoter, which normally regulates actin gene expression in rice. RBCS is the 3' transcript terminator region of the rice rubisco gene. Using these components in backbone vector, you are to design a transformation vector containing the MER-A and MER-B genes regulated by appropriate promoters. The vectors should have origins of replication, antibiotic resistance gene for selection in microbes, a herbicide resistance gene for selection in plant cells, and tDNA borders. If possible, MER-A and MER-B should be regulated by different promoters, but you can use the same terminator on different genes. Simply draw this vector, label the components, and you're done.